Thank you guys so much for joining us today to let me talk about my research. I'm really excited to share it with you. And thank you to Andrea and Matt for hosting this webinar and inviting me here today. Okay, so I think we're the first people from PBARC to have spoken in this webinar series. So I just wanted to introduce you to our center, the Daniel K. Inouye U.S. Pacific Basin Agriculture Research Center located in Hilo. And our mission is to conduct research for the development of sustainable agriculture systems and pest management programs in support of Hawaii, the Pacific Basin, and U.S. agriculture. So today we're going to be talking about good nematodes and bad nematodes. So depending on which side you're coming from, you're probably thinking, I didn't know there was another type. But nematodes are very diverse. They're the most abundant multicellular animals on Earth, and there's over 30,000 described species. So they fill every ecological niche on our planet, and many are parasites, plant, insect, or human and animal parasites, but each being very specific. But the majority of them are free-living, and many free-living nematodes live in the marine environment. So we'll start off with the bad nematodes. So if you're a coffee grower out there, you know what I'm talking about. These guys are microscopic, non-segmented roundworms. They have a stylet mouthpart for piercing plant cells, and they cause billions of dollars in damage to agricultural crops worldwide. So how do these small little organisms damage crops? Well, they destroy and weaken the root system. There will be thousands of them invading just one plant. This will interfere with the uptake of water and nutrients, which cause stunting of the plant, weakens the tree's immune system, and results in poor crop yields. So what can we do about them? Well, there's many different approaches. Some are prevention, cultural practices, host plant resistance, bionematicides, and chemical nematicides. I have one slide on prevention because I think that's the most important. And then we'll be talking a little about host plant resistance. Next time I'm invited to this webinar, we'll be talking about chemical nematicides. There's a couple new nematicides on the market that I've been asked to screen. So we're just getting those experiments up and running now. So I always have to say this, but if you don't have nematodes, these bad nematodes, don't get them. It's really important. If it's clean, stay clean. So the most important thing for coffee is to propagate your plant material by seeds, cuttings, or tissue culture. And that means no pula-pulas. So I know it's very easy to pull up these little seedlings that you see in your coffee orchard, and there's tons of them. But each one of those are most likely infested with the nematodes already. So you'll be starting any new plantings if you use these by having a nematode infestation. So also, if you're going to be growing your own seeds in the nursery, you need to use sterile potting media, grow them out on clean benches. Don't use the same tools in the field that you would use in your nursery setting, and make sure you always clean your pots if you're reusing them. It's very important to have good sanitation practices and train your staff that there are these invisible worms in the soil that we don't want to spread around. And it's good to watch the water movement from infested fields. And sometimes that's the way the nematodes will travel. They love surfing on the water, and I bet a lot moved during this Kona low we just had. So if you are underneath an infested farm, unfortunately, that can cause some contamination in your orchard. So we'll be focused on the Kona coffee root knot nematode, Meloidogyne konaensis. And this is a sedentary endoparasite, which means the female will establish a feeding site in the root and stay sedentary and feed until she can produce her egg mass. This particular species is highly pathogenic on coffee. It's the worst of all the coffee root knot nematodes. It destroys the root system, and it will cause severe tree decline in susceptible Arabica trees within 10 years. So some of the symptoms of a nematode infestation are flagging and yellowing of the leaves, overbearing dieback, a corky taproot. You'll see very few feeder roots, and over time we'll have decline and eventually death of the plant. But the number one symptom above ground is stump wobbliness. So if you can walk up to your tree and you can shake it, and it moves around. And if you can even uproot it by hand, that's definitely a sign of a severe nematode infestation. And this is what a nematode-infested field in late decline looks like from the aerial view. A lot of times the nematodes will be clustered, so you will see these patchy areas of declining or dead trees. I'd like to introduce Landon Wong. He's a PhD student in nematology at the University of Hawaii at Manoa. And he's focusing his dissertation research on the taxonomy of Meloidogyne conaensis, the Kona coffee root knot nematode. So he's doing genetic work as well as morphological examinations. And he wants to characterize M. conaensis because very little is known about it. He's done cytogenetic analysis, which is counting the chromosome numbers. He's done isozyme analysis. He's also done sequenced primer arrays, so he can make phylogenetic trees from these genomic and mitochondrial sequences. And not to give away the end of the story, but if you look at the top, you can see how very closely related Meloidogyne paranaensis and Meloidogyne conaensis are. Meloidogyne paranaensis is the piranha coffee root knot nematode from Brazil, which has symptoms very, very similar to the symptoms we see here in Kona. So he had to go on a trip to Virginia Tech, and he worked with a specialist who just does root knot nematode morphology. So he got to work with the scanning electron microscope, and he got these amazing photographs of these very, very tiny creatures. And what else is so cool is that he actually extracted the stylet from the nematode, which is the feeding structure, as I mentioned earlier. And got these photos. And when you're trying to identify root knot nematode species, it's very, this is one of the characteristics that you look at, and that would be the protrusions on the stylet. And another way that you can identify root knot nematode species is from female perennial patterns. And to do this, you have to cut the bottom half of the female, which you saw in the other picture was that round globular circle, and then place it on a slide. And then you can actually match these up and find out which species it is by looking at these slides. So the results of his work so far have been super interesting. He has confirmed without a doubt that the morphology, the biochemicals, and the gene sequences are all the same between our nematode and the nematode from Brazil. So he just completed a paper where he was asking the taxonomy community to synonymize these two species as one. And because conaensis was described first by Dr. Donald Schmidt from the University of Hawaii, conaensis will be the true name, and paranaensis will just be a synonym. So this was likely introduced to Kona through infested coffee seedlings brought from Brazil in the 1800s to establish the first coffee plantations. So it's been around for a while, and that's why the majority of coffee plantations in Kona have this infestation. And then after the end of the sugarcane era, coffee growers popped up in Ka'u, and this was in the early 2000s. So they took infested coffee seedlings, the pula-pulas from Kona, to establish these new plantations. It was spread further in Ka'u when the growers used contaminated field soil to grow their nursery plants and sell them to other growers so they could establish new farms. So now, unfortunately, it's pretty widespread in Ka'u as well. So this is a lesson that we all have to remember. We don't want it, to spread any further. And we have done some surveys. We haven't found it in Ka'u, in Hamakua or Puna, and we also haven't found it on the other islands. But there is a very strict quarantine in place for moon coffee around. So we shouldn't get it there if everybody continues following the rules. So what can we do about this? Well, the number one best thing you can do is use grafting as a management tool. So when you graft nematode resistant or tolerant rootstocks, this is a very effective management strategy to plant nematode susceptible cultivars like Conotypica. And also the few CLR resistant cultivars I've tested so far have shown susceptibility. So we recommend grafting as well for those. So with nematode resistant rootstocks, you prevent root knot nematode reproduction. but tolerant rootstocks hold up in the presence of high populations of nematodes due to increased vigor. So nematode feeding will stimulate new root production, and both are great, just graft. So there was a study done, 13-year field study, in cooperation with the Manoa CTAR and the Kona Experiment Station team, led by Andrea Calbata. And this tested 8 rootstocks in a field heavily infested with the nematode. And at the final year of the trial, we saw that non-grafted Kona typica had an 81% mortality and a yield of less than 1 kilogram of cherry per tree. And the one that was the most promising was a Liberica arnoldiana, which had no mortality and a very impressive yield of 10 kilos of cherry per tree. That arnoldiana cultivar was renamed Meisner. And this bulletin, extension bulletin, was released to the public to announce the new name. And then this material is now available through the Kona Experiment Station. And Mark Meisner was the farm manager at the Kona Experiment Station for many years, and he was crucial in keeping all of this germplasm alive. And so it's a great honor that we have this to represent his legacy. So you can go to the CTAR Free Pubs website and pick up a copy of this bulletin yourself. And talking about CTAR Free Pubs, please check out these other 2 extension bulletins. One is a pictorial guide to coffee grafting, so you can do grafting at your own farm. And the other explains how to sample for the root knot nematodes with a nice picture and explanation. So one last point on this coffee root knot nematode is just keep in mind that healthy plants are more tolerant to nematode infestation. And with optimal growing conditions, plants are better equipped to fight off plant parasitic nematodes. So optimize your fertilization and soil health and control insect pests and diseases. Okay, now the bad guys, or the good guys, sorry. But they are really, really bad for insects, which is great for us. And this is like using nature to fight nature. So insect parasitic nematodes are microscopic, non-segmented roundworms. But they attack insect hosts and very specific to insects. They do have a broad host range and they're easy to apply. They're safe for humans, animals, and plants. They're not regulated by the Environmental Protection Agency or the Hawaii Department of Agriculture and Biosecurity, and there's no re-entry period or personal protective equipment required to apply these organisms. So how do these nematodes control insect pests? The infective juveniles enter into an insect host. They release a symbiotic bacteria which kills the insect, and then the nematodes begin to develop, reproduce, and then the next generation of infective juveniles emerge, and the cycle will continue. So we did a survey back in about 15 years ago, where we followed in the footsteps of Dr. Arnold Hara, who is an extension specialist at CTAR. And he went all over the Hawaiian Islands looking for these insect parasitic nematodes, which we also call entomopathogenic EPNs. And we followed in his footsteps, and we were able to recover this species Heterorhabditis indica on all the islands. And this is a picture of Kathy Mello and her husband who helped out with the survey. And they got to visit a bunch of really amazing beaches across the state because that's where these nematodes were recovered. So we cannot bring in any commercial EPN products. So that's why it's great that we have these locally isolated ones that we're able to utilize for insect pest control. And this particular species is great because it's highly virulent against a broad host range. It's an active hunter, so it cruises through the soil following chemical cues from its prey. And we've tested this against 40 different invasive insect pests, including small hive beetle, banana moth, coffee berry borer. One of the most successful ones that we've used it against is sweet potato weevil. So the nematode can actually travel through the soil into the sweet potato tubers, hunt down these sweet potato weevil larvae, reproduce in the tubers, and then come out to the soil and move into the next tuber. So we're excited about the success with that. And we're currently doing studies with two-line spittlebug. But today we're going to talk about Queensland longhorn beetle, which is a large large wood-boring cerambycid with a very wide host range. And the larvae cause extensive structural damage. And these are some of the symptoms on cacao. You'll see the sawdust-like frass, the oozing sap, the lifting of the bark where the larvae were feeding underneath, and girdling of the trunk. You also will recognize these adult emergent holes, which are large round circles, and then that is the end of the life cycle of the larvae in the tree, and that's when the adult will come out, mate, and begin to lay eggs. And this is pretty seasonal. Most of this occurs within a few months over the late spring, early summer period. So QLB is very damaging to orchard crops. It has a wide host range. The larvae will create galleries within the tree, and they'll girdle branches, so you'll have like whole branches falling off. But once the large larvae tunnel into the middle and girdle the trunk, that's when you're on the point of no return. And I also see this a lot in combination with feral pig damage. So at that point, the trees won't be able to recover. So that's why it's important to scout your orchard carefully and treat it early. So this is a picture citrus, kukui, and cacao. And kukui and breadfruit are the top two favorites, but it's starting to find new hosts all the time. So how we do this nematode application is we use a disposable plastic syringe, and we actually inject a liquid solution of the nematodes, which is just nematodes and water, directly into these galleries that the larvae have created. So important thing when you're doing a nematode application is to store the nematodes in the dark. Use them as soon as possible. Don't refrigerate them because they like it warm. But they are susceptible to UV light and desiccation. So it's best to apply the nematodes early in the morning, at dusk, or on rainy days. We've had our most successful applications when we're getting soaking wet, but the nematodes love it. Important thing to remember is always agitate the nematode solution before transferring it or drawing up the liquid into the syringe because the nematodes can settle. So step one, before you even get the nematodes, go out and scout your orchard. And I mean looking up in the branches, look all around from every different angle. And a lot of times I will first look right at the base of the trunk of the tree because you will see that frass and sawdust kind of falling out from the branches and down to the bottom. So that's kind of a first quick check. And so here's Kathy showing how to scout the orchard. See all of the frass coming out. And this spot right here that she's mashing is very spongy because it's hollow underneath because the larvae have just been feeding everything inside there. So that is a good place to apply the nematodes. So she will mark the branch. So when you come back with the nematodes, it's really easy to find. And then so you'll go back to these holes, you'll inject this needle into these soft spots, and then you know you're delivering the nematodes right to where they need to be. So same thing, she's going to go and try to move to every spot that she can get the needle inserted. And so as soon as you start to see some of the liquid coming out, then you need to, move to the next spot and just try to do as many applications as possible and get as many nematodes in there as you can. So the first field trial we did, this is the result 4 months after one application. You see it kind of drying up. There's no more sap, no more frass, and then you start to see some regrowth of the tree. Another field observation, we did 2 EPN applications. We saw 75% of infested avocado trees didn't show any new signs of QLB activity. In cacao, we saw 50% of no activity after one EPN application, but we never could get the rest under control that season. And that's because the infestation was very severe prior to starting the nematode application, so treat as early as possible. One, because the larvae are very small, so the nematodes can easily overcome their immune system. And secondly, the larvae are closer to the surface. When they're small, the egg is laid just underneath the bark. And so once they get larger, they start tunneling deeper inside. So this program has been really stakeholder focused. We've done a lot of outreach to growers in person and online and trying to train them how to do their own applications. We've worked with a lot of grower groups. and we partner with the Big Island Invasive Species Council to assist us with the EPN rearing and distribution. One bottleneck is ramping up production of the EPNs. We're currently renovating a portable trailer at the Waikea Experiment Station just for this purpose, to increase our host insect rearing and nematode rearing. So if you're a grower on the east side and you have this QLB problem, you can contact Jade or Darcy at the Big Island Invasive Species Council and get a starter kit of these nematodes and the syringe, and this is provided at no charge to the community. So thank you to all the nematode team, my collaborators, and thanks again to Andrea and Matt for being great collaborators and for hosting this webinar so we can get our information out to the growers.